Hello, everybody. This is Dr. Adam Rinde, and welcome to this informational session on Nutrigenomics. If you are in the chat room and you feel comfortable with it, just send a chat message and a quick hello, letting me know that you are here. I do have a sense of how many people are going to be joining us today, and we're going to get started with the podcast regardless, but I just want to give a few moments for people to file in. And this will give me an opportunity to just kind of go over the general emphasis for this particular informational session, and then also some ideas of how to get the most out of being here. First of all, this is not a webinar, meaning it's not going to cover the deep details of nutrigenomics, except it's a overview of nutrigenomics and to specifically talk about a test I use called IntellxDNA. And I encourage you to ask questions as we go, even though there will be time for Q&A. Some of the questions that come up while we're talking will be helpful, and if I feel like they weave into where I'm at in the presentation, I will stop and answer the questions. Most of the questions I'll be able to post so that they show on the screen, or they at least will show in the chat to all of you. And then from there, we can have a discussion about those questions as we go. So if my sound sounds okay to you, can you please just send a quick message in the chat, just letting me know that you can hear me okay, and that the screen is showing the slides. Okay, assuming that we're good to go. Great. Assuming that we're good to go, I just wanted to again emphasize that as the questions come up throughout the presentation, just put them in the chat. And if it's a good time for me to stop and take questions, I will do that. I think it's probably not going to be something that saving all the questions to the end would be helpful for the listeners. So we'll stop at different junctures. I'm hoping that we'll start with an overview of nutrigenomics and the emphasis that they play in overall health and in addressing health. We'll go through some details of what nutrigenomics are and what we're actually learning from them and how they apply to specific health conditions. And then I'm hoping to drop out of the slideshow a little bit and share an actual sample report of the IntellxDNA report, and then break for Q&A, and then wrap up. The conversation should last anywhere between 40 and 60 minutes. So without further ado, I'll get going. So I first want to just start with an overview of how genetics may play a role as a driver in disease. Well, that's pretty obvious that for most, I think that genetics are studied as an underlying root cause, and most people sort of shrug their head and say, well, what am I going to do about genetics? Well, I think through this conversation, you'll learn more about this concept of epigenetics and how we can strive to influence our predictors of health, such as genetics. But I want to just point out here that this particular diagram I use a lot in my study of how I look at health. And on the bottom floor are various genetic factors that we think about that might kickstart a disease process if they are turned on or are are active. meaning we have different genes in our body that control or regulate inflammation, detoxification, autophagy, coagulation, nutrient receptors, neurotransmitter production, hormone function, and the list goes on and on and on. And once those genetic factors are being pushed on by certain environmental pressures internally and externally, then they start to manifest with disease processes such as metabolic dysfunction, oxidative stress, tissue repair issues, depletion of antioxidants, nutrient deficiencies, toxin overload. And so, as those processes begin, we start to see people develop diseases. And what we're thinking about in the lens of this discussion is understanding what's going on below the water, below the iceberg, before it manifests as a condition that actually is a disease that needs to be managed. So, by looking underneath, we can see certain predictors that may lead us to having these diseases. The goal, again, of health, in my kind of view, and in a lot of people's view, is to live as long and healthy without a major disease. And when and if that major disease manifests, that period of the lifespan is a very short period. And so understanding these below the surface dysfunctions can mitigate and help us prepare and delay the onset of major disease. So that is kind of like the overview of how to look at this. Now, epigenetics is a term that will come up in this topic of genetics. This is looking at everything that is more under our influence or is created in the environment that puts pressures on the layer of genetics. I've put layer one, the inside of this onion, as being the genetics, and everything that's atop this layer as being more epigenetic layers, things that we need to be thinking about that are putting pressure on our genes and driving possible expressions of gene dysfunction. So nobody's born with a perfect set of genes, but the more layers that these environmental pressures put on the genes, the harder the gene processes have to work, and the more likely dysfunction is going to appear. This is not something that has been a proven model to look at as far as, which layer is the top layer, which layer is the second layer. This is just my current view of the things that we need to kind of peel back to help our genes. So layer 6, the outside layer, and I also think many of these layers kind of go in order of things that we have most control over and most influence over in our lives. So layer 6 being sleep, exercise, and diet, the most accessible epigenetic factor. And so we can't lose grasp on the fact that those are number one. Then layer below that, gut and digestive health, another major influencer on gene health and gene expression, and on the amount that the gene factors need to work, because these genes are influenced by inflammation and immune system events, which 70% of the immune system is in the digestive tract. 90% of our serotonin is produced there. And it's a major gateway of inflammation entering the body. So in my view, gut and digestive health is layer 5. Layer 4 will be environmental toxicity. Not only can toxins damage DNA, but they also add more inflammation and oxidative stress into the body, changing the way that our genes can function, making genes work harder and putting pressure on detoxification pathways. And this is another epigenetic pressure. Some people might think that's the top pressure. Again, I'm going in order of what I feel people have more control over. Layer 3 is hormonal health and mitochondrial health. So, as we're getting closer to looking at these systems, mitochondria is obviously the internal energy source and the controller of the life of a cell. And so if mitochondria is damaged or mitochondrial is overly stressed or not functioning well, we tend to see more oxidative stress and inflammation in the body. Hormonal health plays another role. Hormones are important for counter regulatory actions, for inflammation, for neurotransmitter production, for helping combat oxidative stress. So, as hormonal pressures or hormonal layers are not being addressed, we'll see again more pressure on the genes. Layer 2, spiritual emotion and emotional health. There is a lot of discussion, such as with uh, professors and researchers like Bruce Lipton about the impact of our mental and spiritual health on our genetic outcomes. And we can't deny the impact of stress and the impact of depression and anxiety and other hypervigilant states on the inflammation in our body and the oxidative stress and our overall regulatory systems. So that to me is an internal layer that's very close and an important thing to discuss with epigenetics. So that kind of gives you the big picture view. And again, feel free to ask any questions as we go. So we're going to get into some nitty-gritty detail now of the genome and what it is. So some terms to know: the whole collection of your DNA is called your genome, and it's the code, considered the code of life for our bodies and code of our predictors of our health outcomes, responsible for many functions and transcriptions of proteins. And so the term genome refers to the entire DNA collection. So our DNA is organized into 23 pairs of chromosomes. These chromosomes carry genetic information, and these are what we're studying when we're looking at the nutrigenomic panels. This is something that describes the code that DNA has. So much like the computer programmers use ones and zeros to program code for software, the DNA is made up of these different base pairs of proteins, essentially guanine, cytosine, adenine, and thymine. And the DNA is coded with different configurations of these base pairs to give us our individual blueprint in our body. And so the differences in one human to the next are often based on the organization of these base pairs and how they laid down during development. So we have 25,000 protein coding genes and many more regulatory genes. So to understand what these genes do and how they play a role in health, you can see that many genes are transporters for vitamins. So you can have issues with vitamins such as B12 or B6 or vitamin D that are based on genetic coding, and some people have more efficient genes for those transporters, and others have less efficient. Genes code for the tissue and arteries, so we can see arterial defects being influenced and the impact of arterial damage being influenced by genetics. Repair of tissue is coded by genes, so people with more chronic inflammatory conditions, such as osteoarthritis, can see that some people are more coded to more favorable response to repair and regeneration, and others are not. Inflammatory signals are coded by genes, so some people have a tendency to have higher cytokines and higher response to infection, or more predisposition to autoimmune disease because of these decisions that are made in our development. Brain chemicals, so some people have better transmission of dopamine, serotonin, norepinephrine, histamine based on coding. And our detoxification is highly influenced by this. So some people that have been called like canary in the coal mine, people who get set off by very little chemical exposure, may have polymorphisms or genetic changes that make them have less of ability to detoxify certain chemicals and toxins or exposures, whereas the next person might walk into a highly toxic environment and have stellar detox genes and be able to tolerate a major chemical exposure with no issue. So this is just another view of what I talked about, how you can see the DNA is laying down these different base pairs, and a SNP, which is single nucleotide polymorphism, is really at the heart of what we're studying when we're looking at genes. So a polymorphism is either what's called the wild type, which is the normal polymorphism that does not express with increased risk of problems with the gene, or there's a polymorphism where we see a change in a base pair that may be one small change or a couple small changes that end up changing the function of that gene. So these variants can show up in whether we have brown eyes or blue eyes. They are essentially like one ingredient that could change the recipe. By one base pair change, it could change the outcome and view of essentially how our body expresses. Okay, I think we're back. There was a little bit of blockage in. Okay. Okay. So sorry for that interruption. There was just a little bit of a network blip. As I was mentioning, there are some polymorphisms that have to do with nutrients. Test, 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 test. Okay. Can you all hear me okay? Okay. Um, I would just refresh your screen if you can't hear me and then jump back into the session because apparently it looks like I'm good to go. Okay, great. Everybody's good. Sorry about that. There was a network blip here on my end, and I think we're good to go. So as I was mentioning, the B12 in some people might not be transported properly, and the tests that we do can identify if that's a transporter issue. And some people might just need higher amounts of B12 to actually get enough B12 into the system and transport enough. That can be a transport issue or a receptor issue, and we can identify why someone might be having chronic macrocytic anemia, or possibly more issues with neuropathy or more issues with depression or anxiety because of low B12. So a lot of this is redundant, but while we identify these SNPs, or some people call them variants, um, we can understand what might be beneath the surface of why people are not feeling well. Using this information is only as useful as knowing what to do about it. And one of the things that I like about nutrigenomic testing is that it's studying how nutrients and lifestyle moves can influence these genetics and help them function better. Um, I think some of the genetic testing that's out there may show what you have risk for, or might kind of talk about sort of your overall genetic traits or your genetic risk, but by not able to kind of guide you towards how to mitigate these risks, it's somewhat daunting and problematic. Even taking reports to your doctor that shows that you have increased risk of Alzheimer's disease might lead to just sort of a shrugged shoulder conversation. So when I was getting into this, I was looking for more of a solution-based test. So there's a couple terms that you want to be familiar with to understand the deeper discussions. One's called heterozygous and one's called homozygous. This is really important because a heterozygous variation in a genetic test, when we're talking about a SNP, is in most cases less problematic than a homozygous variation, where um, there's two changes on that polymorphism that confer increased risk of a dysfunction with that gene. So understanding whether I have a heterozygous SNP or a homozygous SNP is a very important aspect of the discussion. Also, it's important to know, and this is something that really changed me, is not all SNPs are significant. It depends on where they're located. So when looking at DNA, there's a lot of DNA that has no effect, DNA portions of the strand that has no effect on any function. We don't know what that DNA does. And then there's a coding region where we're looking at changes in the amino acid sequence, as we talked about, and how that might in fact affect the coding of proteins. And then there's another area part called non-coding SNP, which changes the amount of the proteins that it's coding for produced. So it's either changes in protein function and sequence or the amount of protein produced. These are called causative SNPs. This is the area of the gene that we actually know that it's related to health outcomes. So person one, we can say versus in person, three people, and they're looking at their DNA, looking at whether a person is more at risk for disease or a person is more showing more favorable signs towards health. Could be as much as one single base pair switch between the person. So person one with the base pair A might have a normal protein in that particular sequence. Person two may have a lower or lower functioning protein on the same protein. Person three with the C base pair could have a higher risk of disease based on that polymorphism. So we're looking at all these different variations when studying nutrigenomics. So this is just kind of shows you, and I'm not going to go through this in too detail, is that the DNA was identified in 1953, the helix structure of the DNA, and in 2003, genomic sequencing started, and we're started to see more nutrigenomics come on the scene up until today, where we're starting to make decisions on medications, on cancer therapies, on all types of pharmaceuticals, such as anxiety medications, depression medications, ADHD medications, and of course, nutrigenomics to mitigate risk factors of disease. We're still very early in these stages, but this is kind of the timeline. So let's talk about some specific areas. And I'm going to pause. And again, if you have any questions that are coming up so far, please let me know. So when I'm looking at using the IntellxDNA test, one of the things I like about it is that it breaks down different real useful categories to look at, and it has panels that are designed based on common concerns amongst patients. The other thing that I will say about the IntellxDNA approach is a lot different than I was trained in nutrigenomics. So when I was first starting out in nutrigenomics, most of the information out there was based on common polymorphisms that were seen across large populations, and so we would tend to discuss polymorphisms that were in greater than 30 to 50% of the population. And when I started working with IntellxDNA, I realized that their approach is completely different, and they look at more rare polymorphisms that show up in a smaller part of the population that are more associated with risk of disease or risk of pathologic or pathophysiologic changes. And so what made sense to me is that if there's a gene that's more common in the population and this polymorphism is being discussed, then it likely wouldn't be causing much problem in our populations if it's been passed on for so many generations and hasn't been kicked out of it hasn't been kicked out of reproduction and passing the genes on to future generations. What made more sense to me is the rare, unusual polymorphisms that are researched and are predictive of disease, because those are going to be more individualized to the person that we're talking about and the person that's ordering this test. So one of the genes that's studied in the stress component is called COMT, and this one is a gene that's involved with catecholamine um, uh, breakdown. And when it is slow, and there's a homozygous polymorphism on this, then we tend to see a lot of adrenaline built up, high amounts of cortisol, flight or flight reactions. This is a gene that's linked greatly to cortisol and stress response and studied a lot in the literature. It's also involved with detoxification and hormone detoxification. So we'll see that some people will come back with a extremely high 3 to 4-fold levels of cortisol based on having a polymorphism in COMT. And so this is people that are more prone to panic attacks, anxiety, stress response. Um, this also is a superpower for some people. So we can't look at these are like good or bad. So there's a benefit of having slow COMT too. You tend to be very high attention to details, you tend to find problems, the needle in the haystack. You tend to be very responsive to danger, as far as being able to be aware of danger. And so this is one area that Nutrigenomics looks at. And like I said, there's good news when we find these polymorphisms, what to do about high cortisol. Um, there's important reason to address this. But, the other thing that COMT has with it is increased risk of heart attack, 1.7 times risk with 2 copies of COMT compared to those without. And also, people with COMT polymorphisms tend to have elevated homocysteine, which is involved with being cardiotoxic, neurotoxic type of compound. So very higher risk of 2.94 times risk of higher levels of homocysteine. So this is an important thing to know because COMT is very involved with methylation, and homocysteine is a marker of poor methylation. So if that pathway gets plugged up by slow activity, then we can see high levels of homocysteine. Good news is we do have several tools in our tool chest that can address a slow COMT. One of them is one of my favorite herbs called ashwagandha, and it tends to help act on this gene. I will say that the literature for the IntellxDNA test that I do is largely based on human studies. There are some based on animal studies just because we're way behind being able to research everything out there. But I think it's just really important to know that all the recommendations are based on some type of mechanistic study that was done. And in the report, all the references are available and can be cross-checked. And sort of ranked by evidence as well. So this is something that if someone came back with a slow COMT, this is one of the many things that might be discussed. Also, just as a side note, B6 and magnesium play a big role in that. MTHFR, lots of people know about this particular polymorphism. This one is involved with methylation. People who have issues with MTHFR tend to have problems with making active folate, and there tends to be a lot of downstream problems from that, anything from neurotransmitter issues to depression, cardiovascular inflammation, increased risk of stroke, hearing loss, fertility issues. It is talked about so much, and to know kind of the variation, there's a big difference between being heterozygous and homozygous as far as concern. And there's a lot of ways to overcome having a methylation issue. One of the tools that sometimes is recommended if people have what's called a homozygous MTHFR polymorphism can be a form of folic acid called L-methylfolate. It's shown some really good data in helping address depression. Makes sense because if you're able to give the body active folate, then that's a key step in helping production of serotonin and dopamine. And, we would likely see an improved response once the cells have that nutrient. Also, increased risk of miscarriage with MTHFR mutations. So a lot of the prenatal supplements are now just coming preloaded with the active form of folate and some methyl cofactors to help with carrying pregnancy all the way to term. Now, this is where it gets interesting is we spent a lot of time studying the kind of single view of these particular polymorphisms. So we would say, like, what does MTHFR mean as an outcome? What IntellxDNA does is it looks at the genes network. So combinations of MTHFR plus other polymorphisms and discuss the connections between these genes. And so there are clotting genes that the Intelix looks at, and having MTHFR plus other clotting SNPs would be more of a risk for clotting or stroke than just saying looking at MTHFR in a vacuum at itself. And so we're kind of compounding the network of genes on this study so we can really understand the big picture. Clotting's a big deal. Obviously, it affects pregnancy, travel, older age, and the Intelix report looks deeply into clotting SNPs. One of the SNPs it looks at is factor V Leiden, and this is a SNP that would increase the risk of stroke and blood clots. And so not only do they look at this pathway in detail, but they'll look at what can mitigate having this polymorphism, what could kind of lower its expression, and things to do to help mitigate the risk of clotting. And also the difference between platelets and excess platelets and fibrin is really important to understand. So fibrin is another blood product that increases the risk of clumping and clotting. And looking at fibrin genetics and risks of fibrin is very important. One of those that's really common that is talked a lot about is called LP(a), and that particular polymorphism, if it's present, means that there is a chance that there's more fibrin in the blood, which is involved with a lot of blood flow issues, nitric oxide issues, and risk of clotting. And so we've learned lots of different tools to help bust up fibrin and help people deal with high fibrin scenarios. Genomics also looks at risks for obesity, bone density issues, neurodiversity, such as autism, ADHD. It's important to know that when studying nutrigenomics, having a functional medicine view is very helpful. If you aren't familiar with Dr. Bredesen's work on Alzheimer's, he's broken down the different pillars of cognitive decline, and to be able to look at these pillars from a genetic perspective is really helpful, because there's a lot of information about what to do about preventing Alzheimer's based on these pillars. So first, I'm just going to jump on the Alzheimer's portion of this slide first. There's genetics that look at risk for neurodegeneration, plaque, amyloid plaque buildup, inflammation that's related to neuroinflammation, mitochondrial decline that tends to lead to oxidative stress, issues with detoxification that can cause neurotoxicity and mitochondrial toxicity, hormone imbalances that could lead to progression of cognitive decline, and vitamin and nutrient transport issues. And then with the cardiac SNPs, looking at all these factors listed here, some of them, when we first started, it was like looking at APOE4 for both cardiac and Alzheimer's. This goes way deeper than that, and looks at these individual factors like risk for calcification, risk for higher cholesterol, risk for oxidative stress. So you can really tease out and have like a functional approach to addressing these issues or preventing these issues. Let's talk about some cases. So just a little time check here. We're closing out on some of the main part of the presentation, and we'll leave time for Q&A. And I actually want to share a sample report. So this is a case of Donald, 51-year-old active male who was coming in for an annual executive physical. He's concerned about his strong history of family disease. He's starting to slow down his running a little bit. He had a normal cardiovascular exam and just gotten back his genomics. And it just shows that he had 6 of the 10 top genetic risk factors for heart disease, and some that doubled his risk of heart disease. So these would not have shown up on a lab. These are below the surface risks. And he, did not have any indication that this was going on. And they decided through the cardiologist that it was important for him, based on this high risk of disease, to go into the cardiac cath lab, and it actually showed that he was on his way to having a heart attack if it hadn't been addressed earlier. So that's like an extreme example when they saw a silent plaque based on these risk factors, showing that he was one of these people that was more at risk for early heart disease. There's some markers that really indicate that, such as Lp(a), and that was addressed. Triple bypass was done, and then came back for prevention. And so, as I mentioned, Lp(a) is a particular protein that is in our blood that helps break down fibrin. And if you have lots of it, it prevents breaking down fibrin. If you have lots of it, then you're more at risk for having fibrin. Now, I will say this is that it is, while he had a SNP on Lp(a), so by testing his Lp(a) in a blood test, you can see that if the Lp(a) was elevated on a blood test, that this process was likely taking place, and fibrin wasn't being broken down. So these are just some of the pathways that showed up on the calcification and inflammation pathway. And another one that he had high risk for. So the way this works is it shows the yellow color is more of a homozygous, heterozygous SNP. The red color is a homozygous SNP, and the odds ratio, meaning that people with this particular variant has 2 times greater than the general population to have more adhesion and inflammation in the coronary arteries. And so the report makes comments like what medications help reverse this, like statins and specific omega-3s that might help support this pathway. Also, we've learned so much that vitamin K2, a specific form like MK7, can help with reducing atherosclerosis and helping sustain arteries. And that's something that's pointed out on this testing. This is just more information about the importance of K2. Another one that's looked at in cardiovascular genetics is nitric oxide synthase, which is involved with blood flow to the arterial system. So this is a case of Daniel, a lawyer who was having problems with elevated blood sugar, elevated blood pressure, and erectile dysfunction. He's eating more of a paleo diet, weightlifting, and eats a little bit of vegetable. And finding out that he had a polymorphism on nitric oxide synthase, 2 variants of nitric oxide synthase pathways, and one of them had almost 4 times risk of erectile dysfunction and 1.3 risk of heart disease, ischemic stroke, and treatment-resistant hypertension. And it tended to affect younger adults, and if obese, the risk increased more. So we talked a little bit about like how to improve nitric oxide production in the body, which foods have high levels of citrulline and arginine is some of the recommendations he was given. It's important to know when you have issues with nitric oxide, it tends to damage this glycocalyx layer that is the protective layer on the arterial wall, and there's more likely of encroachment of plaques and damage and stiffening of the artery walls. And so there's various factors besides nitric oxide that affect the glycocalyx, but when you have less nitric oxide synthase there and more less nitric oxide, then there can be more oxidized LDL and more possible clot formation, clumping of the blood, those types of things. As I mentioned, L-arginine is helpful in helping increase nitric oxide. Also, this compound called pycnogenol is involved with improving nitric oxide. So these are things that this particular patient would be advised to look at. Cognition is also one of the major panels that's studied on IntellxDNA. I've already talked about the Bredesen model, and that IntellxDNA is really designed in their cognitive panels based on the Dale Bredesen approach to addressing cognitive decline. So this is Rachel, and gives you some background on her. She's starting to have some issues at 48 with word finding, feeling stressed and anxious, and frequent migraines. And she noticed with her father worsening of cognition, and she did not have the classic risk factor for Alzheimer's, which is APOE4. I just saw something today from Eric Topol. He put out that if you have 2 copies of APOE4, then it's something like 100. He said in his latest post, 100% risk factor for early Alzheimer's. And so that is something that's gone even up as suspicion. She did not have that. So we're looking at other things that might put her at risk for Alzheimer's or early cognitive decline. So what was found on this particular test is that there was a combination of MTHFR and IL-6 polymorphisms, meaning her body had problems with methylation and increased amount of an inflammatory molecule called IL-6, which causes brain inflammation. And if this was active and kind of going, then we would see that the brain would be more at risk of vascular dementia 3.7 times risk and 2.5 times risk of Alzheimer's. So without, just going APOE4, she would not know her real risk of developing cognitive decline. And so now she has some things to track to see if this is active. For example, this particular MTHFR tracking homocysteine would be really valuable. And that can show if this gene is expressing or not well supported. That's MTHFR. IL-6 can be measured, not that it would be a cost-effective measurement, but it is something that can actually be addressed by certain preventative supplements and dietary moves. And here's a list of things that address MTHFR and IL-6. Green tea is one of the extract of green tea is very popular. Obviously, curcumin, berberine, quercetin, sulforaphane, which comes from broccoli sprouts. These are all things that, with this particular patient, they could be incorporating, or if they could be tracking their levels and seeing at the right time to intervene, or just having a lifestyle thing, but like eating broccoli sprouts on a weekly or twice weekly basis might be a way to mitigate risk. She also had pathways indicated less choline production, other inflammatory pathways. She had a SNP that showed that her estrogen transmission and receptors in the brain were at risk for less estrogen in the brain, which is an independent risk factor for early cognitive decline. And she had an amyloid pathway that showed less breakdown of amyloid. She also had 2 copies of brain-derived neurotropic factor polymorphisms. This is the neuroplasticity gene, and people who have that combination have a 2.7-fold risk of Alzheimer's, and that's almost equivalent to an APOE4 risk. So it's fertilizer for the brain. It keeps the brain learning and synapsing. Low levels lead to Alzheimer risk and are related to depression. This shows the pathways that came back on the report for BDNF. And obviously there's like things to do. High butyrate producing foods increases BDF. Exercise is another motivating factor to get in, like high intensity interval training or cardio or long walks or intense walks. And milk thistle also can act on this pathway. I mentioned high intensity exercise. It's one of the greatest ways to increase BDNF. I've also seen something from Rinde Patrick about the impact of having a sauna, infrared sauna sessions on BDNF. So we use these to help us with decision making. The context of this is really to kind of look what's underneath the surface before there's a problem, and kind of have your eye on the future and kind of what your potential destiny is, so you can help try to change the destiny of your genes or help to slow them. And we also look at these genes to inform decisions on supplements and medications and lifestyle changes. And so the panel is broken down by a number of different pathways. There's a medical panel, there's a mood and mental health panel, there's a panel on neurodevelopment, there's a panel on cognition. And so they're looking at tons of data coming back from your own DNA to try to make these connections. Okay, before I get into this, I'm going to exit the presentation and just show you a report. How's that sound to everybody? Do you want to see a report? Okay. Either I've lulled you to sleep or you're just so engaged that you're not responding. Okay, great. So the report, so let me just change my screen here. Okay, so this is a sample report. One of the things that IntellxDNA does is it has what's called a hotspot summary, which ranks the most clinically significant polymorphisms that came back on a given report for a particular patient. And then it gives you all the information, every pathway that was studied, so you can kind of go through it in nitty-gritty detail. It's a very overwhelming process to get all this data back. So usually what happens is with the provider, they're going to go through the hotspots first and review those, and then have subsequent sessions to go down the path of these individual details. So on hotspot summary, you can see on this particular patient, they'll look at the patient's variants that had a very low prevalence in the population. So this particular variant, protein kinase C alpha, is only prevalent in 0.6% of the population. And so it talks about what this particular gene does from a very high level. And you'll see you can get into a very deep level by going into the individual report. So usually we're looking at these and seeing, some that are much more concerning. So this particular polymorphism, PRKCA, protein kinase C alpha, individuals with this SNP in particular could benefit from diuretics, and then it also gives you potential interventions to mitigate that. Let's go into one that's a little bit more interesting for most people that are watching this. So the polymorphism HLA-DQ25 shows is only prevalent in 1.3% of the population, and individuals with this polymorphism are more likely to have celiac disease, a gluten immune response, so that gluten triggers a autoimmune reaction. And individuals with this SNP are probably need to be screened deeper for celiac disease, and potentially if they have other markers of celiac disease, going in for an endoscopy, and then this person would be guided towards that. Another one that came back on the hotspots, and just 3.8% of the population was a homozygous gene here for glycoprotein 6 platelet. So this is sticky platelets. Individuals with this SNP have stickier platelets and are more likely to have platelet clumping, and increased risk of DVT. Again, that particular patient might be told that they should be on a baby aspirin for life, um, to help mitigate that. More in this hotspot is issues with this particular detox pathway that has problems with transporting mercury and other toxins out of the liver and gut for clearance. So greater risk of mercury toxicity. And there will be different suggestions I'll show you in a second here of what might be done. So you can see the high level kind of gets you started with big rare polymorphisms that showed up, because we're really interested in those things that are individually rare in the person that we're talking to, because the more common polymorphisms might not be even manifesting in this particular patient. So let's go back to the deeper test. So the detailed report. So for example, when we go into the deeper version of the detox pathway, you can see it reports back all the polymorphisms. Some of them are so prevalent that they're either beneficial or not something that's flagged. But going to that one that we talked about, the ATP binding cassette family with the mercury issue, in the detailed part, it talks about the clinical significance. So people with this particular allele might have higher tissue mercury levels than compared to people with no variants. And then it talks about potential interventions. So NAC and sulforaphane and zeolite can reducing mercury levels, and talks about, in this particular patient, they're going to really watch their mercury intake. And then if you really want to get into gene function, it will, excuse me here, it will go into the nitty gritty of the mechanism. So it's an efflux transporter. So it helps eliminate different drugs and toxins, and the transport out of these genes may be affected. And then it even goes deeper, some more research that you can pull up on this particular gene if you really want to get into the nitty gritty details. I'll show you a couple more that are pretty interesting here. So let's go back to the one we pointed out with the coagulation pathway. So this particular patient, as we mentioned, had stickier platelets through this glycoprotein 6 pathway, and only in 3.8% of the population. So this particular patient is 2.3 times more at risk for stickier platelets than the normal variant. And the clinical significance is people with the G allele, right here mentioned, has an increased risk of a venous thrombus event, 2.3 times more risk than the general population. And then, unfortunately, increased risk of fetal loss, 5.1 times the general population. And low dose aspirin targets this pathway. Also, all these natural agents, if people want to get into more detail, like picogenol, bromelain, rutin, curcumin, the list goes on. In my world, we talk a lot about nattokinase and lumbrokinase as helping with platelets and fibrin. So it just decreases platelet aggregation. So it might not be enough for someone with a platelet issue, but certainly is used for other people with fibrin issues. Since a lot of people are interested in the cognition and memory pathway, want to talk about this particular test does test for APOE4, and you can see this particular patient's status is APOE3, E4, and they do not have APOE4 homozygous, but what they would say is um, uh, that they're looking at this particular variant having 3.2 times the risk of this particular variant has increased risk of Alzheimer's disease, 3.2 times the risk of the general population. And so what they tend to do is aggregate the different risk factors and as you go and show whether some genes are favorable in turning down risk and others are, increased risk. And then potential interventions for APOE4, there's a ton of them, being on a keto diet, supplements like lion's mane, and looking at things like hormone replacement therapy and such. So again, lots to cover here. I will say that when you're doing a genetic test, you don't want to just focus on the bad. You also want to focus on good things, because each of these particular panels show favorable genetics too. So if you have beneficial genes, the test will point that out, and maybe having better clearance of toxins may be your genetic blueprint. So it's important when reviewing these tests that you spend time looking at the favorables, the neutrals, and those that are more rare and risky. So I'm just going to kind of scan through this, and this is the medical one, and it talks all about coagulation, diabetes risk, inflammation risk, histamine risk, obesity and weight control, cognition, toxins, brain atrophy, melanoma risk, macular degeneration, and really looks at this stuff in detail. And then there's separate panels that I can show you in a second here. So you can see there are panels that look at brain optimization, mental wellness, and the one we were looking at was more medical. So there's some that go deeper into just the brain focus. And then there's some that look at mental wellness, like attention and focus, depression, anxiety, ADHD, obsession, compulsions. And then there's a neurodevelopmental panel that slices out information related to ADHD, autism disorders, PANDAS, neuropsychiatric disorders. And so when we're finding out like which test to run, we might run different combinations. They do have a thing where you can order part of the test, and then every time you come back to add an additional panel, you can add on, and they just store your DNA. Let's see here. Okay. So I'm going to just go into the Q&A here. Now, I will say to you the things that I didn't cover is that I didn't talk about like ethical issues related to DNA, privacy issues related to DNA. I do know Intelix is very careful, and they do, do not share your DNA with any pharmaceutical companies or anybody outside the organization. It's de-identified, so it's essentially like a number instead of a name, and so that's very reassuring. And there's things like what are we actually doing here? So we're looking at risk factors. We're looking at below the surface issues that might manifest down the road. Your clinician may, if they know how to do this, might take this information and run biomarker tests to see if this is manifesting now. And then, like in the case with the gentleman with the high risk of early cardiovascular disease, it may warrant a referral and further imaging to see if that something's brewing already. For people who actually have one of these conditions that's already showing risk for, and they're struggling getting better with their current therapies, this may help awaken other nutrients that might be helpful. It may also help with like detox pathways that might need to be addressed, other factors that haven't been looked at and could explain why someone's not getting better. So with that, I think I'm all set with my part of the presentation. And the test is a spit test. It's mailed to your home, and then you get the results back in a couple weeks after that. And then in my particular case, I take 2 or 3 sessions to review the data with my patients. So any questions? This would be a good time. I will say that if you are interested in doing the nutrigenomic testing with me, then the best thing to do is set up a brief meet and greet just to kind of talk over cost and what you're looking for, and then setting up the ordering of the test. The meet and greet's like a 15-minute telemed session with me just to kind of coordinate the details and go through the process and help you make a decision. Okay, so if there are no questions, then I'm just going to log off here. So I think thank you so much for attending. I really appreciate you being here. It was good too. You're welcome. It was good to share this information with you. I will say that I've had some people really get a lot of help from running Nutrigenomics tests. This does, as it showed in the beginning, this does not preclude all the other important things that we do for our health. It's just another kind of very important aspect to consider when you're trying to solve your health puzzle and live your best life and live with the greatest amount of longevity and the shortest amount of morbidity and mortality. So without any questions, I think I'll just log off here, and thank you so much for your time, and I hope you're having a nice evening.